Have you ever imagined a world where we could predict exactly when and how a person would get sick? Or better yet, what if we could rewrite the code of life itself, eliminating diseases before birth? This is no longer science fiction. The genetic editing revolution is happening right now, before our eyes, and its implications are far more profound than we can imagine. Imagine holding in your hands a book containing all the instructions to create a human being. Every characteristic, every predisposition, every detail of their existence. Now imagine that you can not only read this book, but also edit it, rewriting entire pages as you wish. This is exactly what science has managed to do by decoding and manipulating the genetic code. Since Watson and Crick discovered the structure of DNA in 1953, scientists have dreamed of the day when we could not only read, but also modify the genetic code. For decades, this dream seemed too distant. It was like having a book written in an alien language, where we could see the symbols but couldn't understand their complete meaning. But then, something revolutionary happened. With the development of CRISPR-Cas9 technology, described as molecular scissors, scientists finally managed to not only read, but also edit DNA with impressive precision. It's as if we had discovered a word processor for the code of life. The magnitude of this discovery is simply astounding. Human DNA contains approximately 3 billion base pairs, forming about 20,000 different genes. Each of these genes carries specific instructions to create and maintain our organism. With new sequencing technologies, we can read this complete code in just a few hours, something that would have taken decades just 20 years ago. But what's truly revolutionary isn't just the reading, it's the editing capability. Using CRISPR and other emerging technologies, scientists have already managed to eliminate genes causing hereditary diseases in human embryos, create pest-resistant plants without using pesticides, develop animals with specific characteristics for medical research, modify immune system cells to fight cancer. In laboratories around the world, researchers have already managed to cure mice of serious genetic diseases, create pigs whose organs are compatible with humans for transplants, and even reverse aging and isolate. Did you know that our DNA shares 98% similarity with chimpanzees, but we also share 60% of our DNA with bananas? This shows how universal and interconnected the code of life is. Even more surprising, only 2% of our DNA codes 4 proteins. The rest, which was once called junk DNA, we now know has crucial functions in gene regulation. A fascinating discovery is that our DNA could, if stretched out, reach to the sun and back 61 times. And even more impressive, if all the DNA from all cells in the human body were placed in a straight line, it would extend for 16.5 billion kilometers. The horizon that opens before us is as vast as it is frightening. In the near future, we could completely eliminate hereditary genetic diseases, increase the human body's resistance to viruses and bacteria, significantly extend life expectancy, create personalized organs for transplants, develop super nutritious crops resistant to climate change. But the possibilities go far beyond medicine. Scientists are already talking about resurrecting extinct species, creating new synthetic organisms to clean up pollution, developing plants that naturally illuminate cities, producing more efficient biological fuels, creating living materials that self-repair. However, with great power comes the ability to edit the code of life raises profound ethical questions. Who should have access to this technology? How far can we go in modifying the human genome? How do we prevent this technology from further increasing social inequality? What's the limit between cure and enhancement? Imagine a world where parents can choose not only their children's sex, but also their height, eye color, athletic ability, or intelligence. This might seem tempting, but the implications are frightening. Could we create a new form of genetic discrimination? A society divided between the genetically modified and the naturals? The risks aren't just ethical. Genetic editing is a powerful technology that, if misused, can have catastrophic consequences. Unintended effects on future generations, accidental creation of new pathogens, disruptions in ecosystems, use of technology for military or terrorist purposes. How do we ensure this technology is used only for the good of humanity? How do we prevent its misuse without impeding scientific progress? While we debate these questions, the revolution continues advancing. Laboratories worldwide are developing personalized cancer treatments, creating more efficient vaccines, fighting rare diseases, increasing food production, seeking solutions to environmental problems. The future is being written now, letter by letter, gene by gene. The question is no longer whether we can edit the code of life, but how we will choose to use this power. We are living in a unique moment in human history. For the first time, we have the power to direct our own evolution. The possibilities are endless, but so are the risks. The code of life has been cracked, and now we need to decide, as a society, what we will do with this knowledge. And what do you think about this? How would you use this technology if you had access to it? Leave your opinion in the comments, and don't forget to subscribe to the channel for more fascinating content about science and technology. Hit the bell icon to not miss any updates about the latest discoveries that are changing the world. If this video made you think, share it with your friends and family. The more people participate in this discussion, the better prepared we'll be to face the challenges and seize the opportunities of this new era of science.